Chromosome differentiation. I thought you took care of that with that other video. Okay, I'll bite. What the heck is it? Why do I care? Make it quick, I'm in a hurry. Well, it's kind of important, you see. You might want to get some popcorn on this one. It's going to take a little while. Chromosome differentiation. It determines what the results will be 9 months after conception. A normal, healthy baby. A monster. A shapeless mass of aborted tissue. Teratomas. Those are tissues and organs found in babies where they don't belong. Now, aren't you glad you popped that corn? Before we start, let's do a little review from the last video. We talked about ontogeny recapitulating phylogeny in the last video. And what that means is that our development through embryogenesis recapitulates how we evolved as a species over eons of time. And if you look at this diagram, you see that in the first early stage of embryonic development, most of those embryos look very, very similar, even though they're quite different in terms of their species, from fish all the way to human. Now, a lot can go wrong as these embryos develop from something that looks more like a fish until something that looks like a human being. A lot of things can go wrong, and all of that has everything to do with cellular differentiation, more specifically, chromosome differentiation. Alright, we talked about this last time. It's a bunch of rubber band with some paper clips, and they are simulating a cross-section of a coiled chromosome. The chromatin fibers are represented by the rubber bands, and they have been condensed or wrapped up by those paper clips, which have something to do with chromatin condensation, which determines whether or not that DNA within those fibers is accessible for gene expression, which is how you create the cells that we have in each of our tissues. Now, we've opened up this piece of chromosome, and it becomes a circle, a beaded circle, like something like you see on the right. What's on the right actually exists. That's a photomicrograph of a structure that was seen under the light microscope at 400x. So these things exist, and it's up to us to determine exactly what they are and their significance. Okay, in this particular slide, we see that the chromatin circle is fully relaxed, the beads are gone, and that looks very similar to what you see on the right, which is an electron micrograph of some DNA that is complex DNA flower, very similar to what you see with the rubber band model. Only that one is at a very high magnification, 125,000x. So these things do exist. Okay, it's up to us as scientists, as people, to try to understand what's going on here. So what I've presented is something I call a two-cut model, which I talked about earlier in the other video. And if you see on the left-hand side, you see two rubber band circles, which should be DNA circles. And in the middle, they're being cut so that a piece of information is being lost. And there's some diagrams, actual photomicrographs and electron micrographs shown below, which are very similar to what you're seeing in these models. So this is the end of the review from the last video, and it's time to go in a lot more detail to explain why this two-cut or fusion model may be very important in terms of chromosome differentiation. Okay, here you see in section A is one of those paddle fan models of rubber bands. It has 5 replicons. There's 4 on the outside that attach to the center replicon. and where they are attached is where the tetrameric base pairing is occurring. So we're going to move, as we go through this video, we're going to move from rubber bands to a diagrammatic representation of these things, and then we're going to go from that to another diagrammatic representation that is much more detailed, so that we can explain exactly how gene differentiation and gene expression may be occurring within these paddle fans. Alright, the two yellow arrows that you see here are pointing to the double origin of replication that is being shared by the middle replicon with the upper replicon, and that's where your tetrameric base pairing is taking place. Okay, you see that same thing in this next set of arrows, and then you see a single arrow, and what you're seeing there is going to become the future master replicon for that entire complex once it differentiates. Then we continue on around to the next set at the bottom, and finally the last set on the left. Now in frame B, you see the two-cut model with the 4 pieces of rubber band that have been cut out, losing information, so that that actually becomes one giant piece of DNA. Now in picture C, what you're seeing is actually, if you'll notice, it's kind of dark red. It's thicker in there in the center than it is in the last frame. That's because a secondary replicon has replicated in there, shown by the yellow ring within the border of that red circle. Those 3 yellow arrows and that lower red arrow show where the inner replicon is base pairing, tetrameric base pairing, with the portion of those outer replicons that have fused with the original inner replicon. I know that's kind of complicated, but it will be made more clear as we go through and get into the more detailed models of this. That red arrow represents a new double origin of replication that will be the master replicon for that entire paddle fan complex. All 4 of those outer pseudo circles and that inner replicon work together to replicate that whole complex, or what I call a replicon cluster. Now those 4 new arrows that point straight up and down and sideways, those are where the master replicon is not base pairing with those pseudo loops, because those loops have lost information in there, and therefore there is only a single double strand in there that is alone, no tetrameric base pairing in that area. Okay, we're going to open up this Replicon cluster so you can get a better look at it. You can now see the master replicon in the center of this complex, as shown by the yellow arrow. The yellow arrows that you now see are pointing to 4 small dimples in the outer circle. Those dimples were tetramerically base paired to the master replicon, as shown by the 4 arrows pointing to it now. In E, as we open up the outer circle even more, notice the remarkable similarity between the inner circle and the one in the electron micrograph in F. A similar comparison can be made between the outer replicon shown in E and what you see in the electron micrograph in F. Okay, we're going to move to a different model now, the fission model, which should be fairly apparent. This is where the replicons, the outer replicons, dissociate from the inner replicon, such that you have 5 separate replicons showing in this particular diagram. So there is no more tetrameric base pairing here. everything is now single origins of replication. Alright, in this next model we're going to combine both the fusion and fission models together into a single replicon cluster that can differentiate by doing both processes, sort of a binary code similar to what you see in computers. Okay, now you'll notice in the picture B that you have three arrows pointing at where the two-cut model has taken place with three of those replicons. So that's the fusion model at work. And then you'll notice that one of those replicons, the fourth one, dissociates from everything and creates the fission model, creating a single origin of replication in the main complex as well as the dissociated replicon. Once this complex is formed, the master replicon gets generated and it creates tetrameric base pairing as shown by those arrows. Two of those tetrameric pairing sites are actually double origins of replication now. The next set of arrows shows the lack of base pairing between the master replicon and those three loops outside that are actually have gaps in there, as we showed before with the fusion model. Okay, we open up this model as we did in the earlier fusion model, where you have your central replicon and your outer replicon, and we're going to compare that again to an electron micrograph. But this time we're going to go a little bit further. We're also going to compare it to other kind of structures that can be found under the light microscope, and you'll notice that all of these structures have one thing in common with what we've been talking about. They have a hub inside of a circle, only these things are much, much larger than what we have been talking about previously. Okay, now this diagram is about stem cell replicon clusters. Now, a stem cell is partially differentiated, but it is still primitive enough that it can replicate itself. And in the process of doing that, it has to not only replicate itself, but to create a daughter replicon, like a daughter cell, but it's a replicon cluster, that has now differentiated so that it can go on and form the tissue type of cells that it's supposed to form. So we're talking about differentiated, we're talking about some part of that chromosome, of that stem cell, part of a chromosome that is undifferentiated, and it will have to go ahead and replicate, and then that one of the daughter cells differentiates. So what we're looking at here in A is an undifferentiated replicon cluster. We're seeing all that tetrameric base pairing that we've seen before. And when it replicates, it reproduces itself. It replicates itself as it shows in the upper arrow in B. But then in B, one of those daughter replicons differentiates as it shows in C. And that involves precocious replication of the inner replicon, like we've talked about this before, creating a double origin. And we're going to talk about these origins in the next slide a little more. In D, the replicons all fuse together, the four slave replicons and the inner replicon, except for the copy of the inner replicon. And so what happens is you produce a replica of the original stem cell replicon cluster on the top and a differentiated replicon cluster on the bottom as shown in E. Okay, we're looking at a diagram regarding bidirectional DNA synthesis. We're going to compare an undifferentiated versus a differentiated replicon cluster in order to help explain the importance of double origins of replication. So if you look in A, this is an undifferentiated replicon cluster. and it has a primary initiation point, that double origin of replication. That is, and as it proceeds, as it initiates at that primary initiation point, the DNA replication goes bidirectionally across that replicon cluster. And it's like a fuse almost. It lights up the other replicon origins of replication, the double origins of replication, as it goes around there from one direction to the next, and then it continues on, as you see in B, to activate those other two double origins of replication, and then it finishes up in C. Now, when you look at D, we're looking at a differentiated replicon cluster, and there is only one initiation point. That's it, because you have a master origin of replication, a double origin of replication, only one of them. The rest of them are gone. And as it starts to undergo bidirectional replication, it follows those slave loops around, as you see in E, and it continues to go around as you see in F. So the point here is that in the undifferentiated replicon cluster, the bidirectional replication is very fast and very quick because there's so many other double origins of replication that can be activated. And this is very important in a process called cleavage during embryogenesis, because cleavage, the cells replicate very, very fast, and they're all undifferentiated cells, so they have to have a means to replicate very fast, and it's because they have all of these double origins of replication available to them that allows them to do this. But as cells start to differentiate, you start losing some of those double origins of replication, like occurs in D, and the ability to replicate starts to slow down because of the lack of these double origins. So we've talked quite a bit about double origins of replication, which involve tetrameric base pairing, but we've not talked about the single origins of replication, which do not involve tetrameric base pairing. And we've already shown that these single origins of replication can be generated by losing replicons, dissociating replicons. Now, mainstream science would say that all bidirectional DNA replication occurs with single origins, not double origins of replication. Not only in eukaryotes, but in most prokaryotes as well. Now, there's only one problem with this kind of thinking, is that where do all these origins of replication go after cleavage is done? These things disappear. They become inactivated. My model explains how that can happen using a master replicon to compensate for the loss of those origins of replication in that differentiated replicon cluster. Okay, we're going to do a quick review here with these rubber bands. We have our undifferentiated replicon cluster showing tetrameric base pairing between 4 outer and 1 inner replicon, and then we also have an origin of replication that's not activated yet because it's still just a single origin of replication. We compare that with a differentiated replicon cluster that has a master replicon as shown by the ring, and it also has the non-base pairing between the outer loops, and then you have your tetrameric base pairing and a fully activated master origin of replication shown by the red arrow. Okay, now we're going to move away from the rubber band models into a diagrammatic model showing the undifferentiated replicon cluster. Tetrameric base pairing between the 4 outer replicons and the inner replicon are shown with the blue arrows, and then the single origin of replication is shown by that single blue arrow. The diagram to the right of the lower differentiated replicon model first shows the lack of pairing between the master replicon and the outer loops, followed by the tetrameric base pairing between the master replicon and the 4 outer dimples. The master origin of replication is shown by the lower red arrow. Now in the diagram to the upper right, things get a bit more interesting with the introduction of 6 smaller replicons. Each of these smaller replicons creates a tetrameric base pairing with a larger replicon, as shown by that small red area. The differentiated replicon model shown below shows both fusion and fission events occurring. The 2 little blue arrows shown at the top of that diagram illustrate the single origins of replication that have been generated by a fission event. The single origin of replication shown on the larger replicon by the single blue arrow indicates something we call an enhancer site, which we will talk about a little later. The fusion event shown by the next blue arrow will become something called a splice site, which we will talk about also later. This third blue arrow is pointing to a promoter and termination of RNA transcription site, which will also be discussed later. The red arrow is pointing to the master origin of replication, which will be responsible for the complete replication of that entire replicon cluster. Okay, this is the mother of all slides in this video. Don't freak out. We were going to go over this very carefully. I was kind of leading you into these diagrams by using the ones that we talked about earlier. So we're going to go over this very carefully so that you'll understand what all of this actually means. Note the small insert at the upper left-hand corner of the larger diagram. That diagram is similar to what you already looked at, and it will be used kind of like a roadmap to explain better what's going on in the larger diagram. Okay, in the larger diagram there are 9 secondary slave replicons. But in order to save space, there's only 6 of them in the smaller diagram. Okay, now the blue arrow you see pointing to that replicon in the main diagram, and then the same one in the upper left-hand corner smaller diagram. Those blue arrows represent just a regular replicon that will, when this thing differentiates, become the master replicon of that entire complex. Okay, these 2 blue arrows are pointing to double origins of replication, of which there are 13 shown in this diagram, along with 13 replicons. Now these replicons are of 3 types. You have your center replicon, which will become the master replicon, and then you have 3 larger loops attached to it, and then and those 3 larger loops are called the primary slave replicons. And then you have 9 smaller replicons attached to those slave replicons, and they are called secondary slave replicons. Okay, now the double origin of replication that you're looking at here is will eventually give rise to something called a splice site if it survives differentiation. And you'll notice it has these little loops sticking out from it. Those loops, double loops, are actually going to become the intron lariat loops once the gene is transcribed and then the mRNA from it is processed from heterogeneous RNA. And finally, you see the double origin of replication that's attached to something called a chromonemata that you see down there at the bottom of this replicon cluster. That is how this replicon cluster attaches to the chromosome. Now a lot has happened here with this differentiated replicon cluster. You'll notice that the 3 primary slave replicons have fused with the master replicon, which has recreated itself so that it can generate a double origin of replication, which will be used to replicate that entire complex. 4 of the secondary slave replicons are gone. They have used fission to be released, and then the remaining 5 have fused with the secondary slave loops to become part of a giant outer circle surrounding the master replicon. Those 2 little blue arrows are pointing to where the tetrameric base pairing is occurring between what is now a giant outer circle and the master replicon. You'll notice that there's kind of a gap between those 2 circles in terms of those little base pairing sticks, but actually that should all be one continuous tetrameric base pairing there. Alright, what these 2 new blue arrows are pointing to, you see a P and a T. The P is the promoter site for transcribing heterogeneous RNA, which will turn into mRNA once it's been processed. The T is the terminal end of that. So you'll see that there's 3 of these things where the slave loops have attached to the master replicon. Okay, the blue arrow you see pointing to the black area on the large diagram, that little black section, that represents what's left over from the fission event when that replicon exited that primary slave replicon. And what you have there is a single origin of replication, which will become an enhancer site for transcription. The next blue arrow in this series is pointing to a splice site, which we will talk about in greater detail later. Okay, what you're looking at here are 3 possible transcriptional pathways, and they are denoted by the little red circles that show the different paths that could be taken with each of these 3 pathways. Alright, in this diagram, section A represents the transcriptional pathway that would be taken in the previous diagram of the loops that were at the top of that other diagram. And you'll notice that you can see the arrows that are going following from left to right around that piece of heterogeneous RNA, and it starts with the blue arrow pointing to the 5' position, and then you can follow the little red circles all around until you finally end at the terminator region denoted by the blue arrow. In section B, those 3 blue arrows are pointing to that part of the heterogeneous RNA that will become the final mRNA. Those next 4 blue arrows are pointing to the intron part of the heterogeneous RNA. These will be excised from the heterogeneous RNA and allow the completion of the mRNA. Those 2 blue arrows are pointing towards the exons, which will eventually fuse with the remainder of the RNA to complete the mRNA. In section C, those intron loops have rotated 180 degrees so that the exons can line up with the remainder of the RNA to complete the processing of the mRNA. The 4 blue arrows show the joints where the exons will be fused with the remainder of the RNA. In section D, the mRNA has been completed, as shown by the big blue arrow, and the smaller double arrows are showing the intron lariat loops that will be released from the RNA. Okay, now we're going to talk about something that's quite a bit different than what we have been talking about in this video. We're going to talk about Dolly the sheep, the animal that was cloned from stem cells or something like stem cells, and you got a complete animal from just a single stem cell, an adult cell. We're going to talk about how that might be able to occur using this model that we've shown in this video. We're going to start by showing a diagrammatic representation of a chromosome, and we're going to include two replicon clusters. Remember earlier in the video when I was using rubber band models with paper clips to illustrate what a replicon cluster might look like if you removed it from somewhere inside the chromosome? That's what these replicon clusters represent. Here is the centromere of that chromosome, and the upper chromosome arm, and the lower chromosome arm, which are called chromatids. Okay, that was the easy part. Now it gets a little trickier. If you look over on the left, the chromosome that has no color in it, but it has the two replicon clusters, those are called, we're going to call them undifferentiated and accessible to gene expression. Now the second diagram shows the lower replicon as being painted black, and so that we're going to call that still undifferentiated, but it is inaccessible. And what we mean by inaccessible means that it has been condensed. The chromatin has been condensed like we showed in that earlier rubber band model with those paper clips, only even more so, even more condensed, and it's inaccessible for any kind of gene expression. Now when we look at the third diagram, at the top you see a green replicon cluster, and that replicon cluster is considered differentiated and accessible for gene expression. Now we're going to look at the chromosomes of a different stem cell, and you'll see the pattern for the replicon clusters is the opposite of what we saw in that first stem cell. We start out with two undifferentiated replicon clusters. But in this stem cell, the undifferentiated, inaccessible replicon cluster is at the top. The differentiated and accessible replicon cluster is on the lower chromosome arm. Okay, now you're going to have to pay really close attention here because it's going to start getting a little tricky. I'm going to introduce you to the concept of de-differentiation, where a differentiated cell reverts to a more primitive state. And an example of that is when you think of amphibians like salamanders. If they lose an arm, they can actually de-differentiate the tissues around the wound, and they will revert back into a primitive form of cell or stem cells that can now regenerate the salamander's arm. Imagine if we figured out how to do that for humans. Well, apparently we've already accomplished that by cloning adult animals from stem cells. The stem cells had to have all of the necessary genes and DNA needed to produce an entire animal from that cell, even though that cell was partially differentiated. If you'll notice in this model that I'm showing you, there is information that's been lost. If you recall the two-cut model, there are pieces of DNA in this model that have been lost. Now, DNA that has been lost is known in the literature in the terms of small circular DNAs. So how can you lose information in stem cells and still be able to reconstitute an adult animal using those partially differentiated cells? That's what we're talking about right here. So we start with the undifferentiated and inaccessible replicon cluster, the black block shown in the chromosome on the left, and we're going to take that thing and we're going to convert it into a undifferentiated and accessible replicon cluster. Now that's all well and good until you realize that we already have a differentiated and accessible replicon cluster in the upper arm of this chromosome. So what we will be creating is a chimera, which will be a cell that has attributes of two different stem cells. And if you have a cell like that, you're going to create a monster. The only solution is to take that differentiated replicon cluster at the top and make it inaccessible in that cell. So that cell only has one function and is no longer functionally a chimera, or two different things connected together. So once you have silenced that Replicon cluster in that upper arm, you are now free to go ahead and activate the lower replicon in order to create the kind of cell that you need for the development of that individual. So your stem cell happily de-differentiates and creates all the tissues needed to create an individual. Well, what happens when it gets to the stem cell that it was originally derived from? What happens to that cell? Well, the stem cell has to go through all the motions that it did in all the other tissues to recreate itself. So it starts out with an undifferentiated and inaccessible replicon cluster. This replicon cluster may attempt to de-differentiate and go back to a primitive state. But as this particular cell goes through the motions of recreating itself, that undifferentiated and accessible replicon cluster will finally become inaccessible again as it attempts to differentiate the upper replicon cluster, which has already been differentiated. I realize this is a lot of information to have to take in such a short period of time, but if you would like more information, you may go to my blog at evolution4.wordpress.com, or you can send me an email for more information at fabernathy@sbcglobal.net. Thanks for watching this video.